in this video the structures of RNA. So basically nucleic acids are differentiated into two types, these one DNA and the second one RNA. So RNA according to types, RNA will be classified into two types. The first one, that is the genetic RNA, and the second one, that is the non-genetic RNA. So in some living organisms, basically those are lower invertebrates or the lower animals like some bacteria, like some viruses. এদের ক্ষেত্রে কি হয় কি ডিএনএ থাকেই না, the DNA is absent, completely absent, and in their cytoplasm basically RNA is present and RNA act as a genetic material. That means RNA দিয়ে এরা ক্যারেক্টারস গুলোকে ট্রান্সফার করে from one generation to the next generation. So that type of RNA is known as genetic RNA. For example, the genetic RNA are found in TMV, that means tobacco mosaic virus, this one, and the second one, that is influenza. Influenza also contain the RNA, no DNA, absent of the DNA. So that type of DNA RNA is known as genetic RNA. And in some cases, these are the non-genetic RNA. Basically, non-genetic RNA again differentiate into three types within a cytoplasm of higher animals. These are the first one, that is the mRNA, M for messenger. The first one, that is the mRNA, M for messenger RNA. And then the second one rRNA, R for ribosomal RNA. And the third one, and this one the tRNA, and T for transfer RNA. So RNA are classified into two types. The first one genetic RNA and the second one non-genetic RNA. In case of genetic RNA, within their cytoplasm, DNA is completely absent, and non-genetic RNA, these are also present within a cytoplasm, basically the higher cells like eukaryotic cells or higher animals or higher vertebrates. And non-genetic RNA, basically examples are tobacco mosaic virus as well as influenza virus. Except these three, mRNA, RNA and tRNA, except these three, and the another RNAs are, those are not important but present within a cytoplasm. These are the first one, that is the snRNA. snRNA is also known as hnRNA. What is the full form of snRNA? That is short nucleated RNA or heteronucleated RNA. This is also the another types. Then the second one, these are called gRNA. What is the meaning of gRNA? G for guide RNA. And the last one, that is miRNA, that means microRNA. So these are the another types. The first one snRNA, SN or hnRNA, that means short nucleated RNA or heteronucleated RNA. Second one gRNA, G for guide RNA. And the third one miRNA, Mi for microRNA. So these are the another types, and also these are the non-genetic. So first one, that is the mRNA, RNA, tRNA. The first one, we notice the structures of the RNA. Basically RNA as well as same to DNA, the basic difference are, normally the basic difference, the first one, that is the pentose sugar. So pentose sugar, that means 5 carbon component, and this 5 carbon component, the first one, that is presence of H, first one, that is the presence of H and OH, and the second one, that is also H and OH, that is the difference between the DNA and RNA, and the third one, that is also presence of H and OH, and the fourth one CH2OH, that is the fifth one, and CH2OH and H. So this one the 1, then 2, then 3, then 4 carbon, and this one the fifth carbon. So as well as DNA, the first time, that is react with the H with the nitrogen bases like either A or G or C, and these are the exception, that is U, because in case of RNA, thymine is absent and it replaced to the uracil. And in fifth positions, there is the presence of same, there is a phosphate, that is a double bond O, then the here also single bond and also O, and this H is replaced to here, that is H plus. So that is the structure, that is the first one pentose sugar, then the nitrogen base, then the phosphate. And the basic difference is this, that type of sugar, basically that type of pentose sugar, basically ribose sugar. So ribose sugar, then the nitrogen bases, and nitrogen bases, the difference is there is a U, and then the phosphate. So 1 mole pentose sugar, 1 mole nitrogen base plus 1 mole phosphate equal to a mononucleotide, and in that case it will be RNA. And the another difference, that is the RNA is also single stranded. RNA is not a double stranded structure, RNA is a single stranded structure. The first one, that is the mRNA, M for messenger. Actually DNA যে পারফর্ম করে, DNA ডাইরেক্ট পারফর্ম করতে পারে না। DNA functions যেটা আমরা বলি কি DNA functions, it synthesize protein, but it cannot directly synthesize. DNA actually help নাই, and the helper are, that is called mRNA, M for messenger, because DNA যা যা বলার আছে, all the informations via mRNA, that means messenger RNA. Then mRNA convert to protein, and mRNA convert to protein, that is the process actually. So DNA have three properties. The first property is DNA will be synthesize its own copy. The process is known as replication. That is called replication. Then the second one, DNA synthesize mRNA. That process is known as transcription. This process is called transcription. And then the third one, that is mRNA convert to protein, and that process is known as translation. So basically these three properties of the DNA are accumulatedly known as, is called central dogma, is accumulatedly known as central dogma. And one MCQ questions, the proposal or the concept provided by Crick, that concept, that central dogma concept provided by Crick, not Watson, only Crick. তো এটা একটা MCQ কোশ্চেন ছিল কি সেন্ট্রাল ডগমা প্রপোজড বাই, অপশনস ছিল ওয়াটসন, ক্রিক, বোথ, বাট অ্যান্সার ইজ ক্রিক, অনলি ক্রিক. Then one explain the first one, that is the mRNA. mRNA basically synthesized from DNA, and at first DNA will be unwind. At first DNA will be unwind. That means that is the structures of the DNA, and DNA will be unwind. That means that is the DNA will be unwind, and that type of conditions is called transcription fork. That means fork means these are the open portions, these are the open region, and any one chain, that is the DNA structure, and any one chain is helpful to synthesize mRNA. Basically যদি আমরা উপরের চেনটাকে নোটিস করি, তাহলে উপরের চেনটাকে নোটিস করলে এখান থেকে একটা mRNA ফর্মেশনস হবে. তাহলে সাপোজ যদি এখানে A থাকে, তো এই রিজিয়নে থাকবে নট T, ইটস উইল রিপ্লেস বাই U. দেন দ্য সেকেন্ড ওয়ান সাপোজ G, in that conditions it will be replaced by C. And then the next one, that is suppose this will be present in T, and then the condition is formations of A. So that is the formations actually. So there is one, the 5 prime, and it will be elongated from 3 prime. তো আমরা যদি এরকম করে নোটিস করে নিই যে mRNA সংশ্লেষটাকে, মানে formations of the mRNA or synthesis of the mRNA, so the total process actually occur into two different types of cells. Suppose the process also occur within the prokaryotic cell, and the process also occur within the eukaryotic cell. Suppose in case of prokaryotic cell, suppose this on the prokaryotic cell, this one prokaryotic cell, and this one the eukaryotic cell. What is the basic difference between the prokaryotic cell and eukaryotic cell? The main difference is that is the nucleus is present and nucleus is absent. In case of prokaryotic cell, that is the naked DNA, that means nucleus is absent. But in case of eukaryotic cell, the nucleus is present, that means DNA will be present within a chromosome. So in case of eukaryotic cell, the DNA will be present within a nucleus, but in case of prokaryotic cell, the DNA, the circular DNA basically, the circular DNA is present within a cytoplasm. তো যখন প্রোক্যারিওটিক সেলের কথা আমরা বলছি, প্রোক্যারিওটিক সেলের ক্ষেত্রে আমরা নোটিস করছি ডিএনএ সাইটোপ্লাজমের মধ্যে আছে, অ্যান্ড দ্য টোটাল প্রসেস, দ্যাট মিনস দ্য প্রসেস অফ ট্রান্সক্রিপশন উইল বি অকার উইদিন এ সাইটোপ্লাজম. দ্যাট মিনস ইন দ্যাট কন্ডিশনস ডিএনএ উইল বি সিনথেসাইজ mRNA, and the total process occur within a cytoplasm. The total process occur within cytoplasm. But in case of eukaryotic cell, the DNA will be present within a nucleus. The DNA will be present within a nucleus. That means at first DNA synthesized mRNA. DNA will be present within a nucleus. That means that this is on the helical DNA, and helical DNA synthesized mRNA. But in that conditions mRNA is not synthesized. mRNA আগে কি হয় কি এখানে কিছু চেঞ্জেস আসে, and that type of all the changes like first one, that is and the 5 prime end, the presence of capping, and the end of the 3 prime end, presence of the tailing, and it covered by a protein, and the protein is called inferrin or informosome. So eukaryotic cells যখন mRNA তৈরি হয়, তো এর সামনের দিকে যে পার্টটা আছে, দ্যাট মিনস 5 প্রাইম পোর্শনস, 5 প্রাইম পোর্শনসের আমরা যদি নিচে একটু বড় করে ডায়াগ্রামটা করি তাহলে ক্লিয়ার হয়ে যাবে. 5 প্রাইম পোর্শনসে আমরা কি নোটিস করি কি প্রেজেন্স অফ ক্যাপিং, দ্যাট মিনস ইটস এ ওয়ান টাইপ অফ প্রোটেকশন ফর 5 প্রাইম এন্ড. And in case of 3 prime portions, that is the presence of tailing. So tailing is also it's a one type of covering, and total DNA, and the middle one is covered by a protein, and the name of the protein is called inferrin or informosome. So that type of RNA is known as A-chain RNA, that is heteronucleated RNA, and these RNA is short-lived RNA. That means the few minutes or few seconds will be survive within a nucleus, but nucleus e ki thake, some pores are present, and these pores are helpful to enter the A-chain RNA within the cytoplasm. And যখন ওটা সাইটোপ্লাজমে চলে আসে, that the capping, these are the tailing, and these are informosomes are not required, because eukaryotic cell নিউক্লিয়াসের মধ্যে একটা এনজাইম থাকে, দ্যাট এনজাইম ইজ কল্ড ক্লিভিং এনজাইম, অ্যান্ড ক্লিভিং এনজাইম ইজ ক্যাপেবল টু কাট দ্য সিকোয়েন্স. তাহলে যদি সিকোয়েন্সগুলোকে কেটে দেয় তাহলে ইটস উইল বি মিউটেটেড, মিউটেশনস হয়ে যাবে, মিউটেশনস হয়ে গেলে পুরো সিকোয়েন্সটা আমার নষ্ট হয়ে যাবে. দেন দ্য অ্যানাদার ওয়ান, দ্যাট ইজ দ্য tRNA, and tRNA is a structures of Cloverleaf model, and this Cloverleaf model Actually Cloverleaf actually scientist, and Cloverleaf explained the structures of the tRNA. So basically tRNA, the structures of the tRNA or tRNA is also known as Cloverleaf model. So Cloverleaf model is structures ta kemni hobe. Suppose this on the 3 prime end, 3 prime end presence of OH, and this on the 5 prime end, and 5 prime end contain NH2. So that is the structures. Because a loop like structures are found, these loop like structures, then a variable part is a variable arm, then another one is called also loop like structures, then the lower portions, and here also a loop like structures. So these are that region is called D loop, D for dihydroxyuracil, that is also called DHU. These are the loop structure. Because why it is loop? Because it blockage. Then this one, that is known as T/U loop. This one is called T/U loop, and that is the variable arm. This one is called variable arm. And the last one, this one is contain presence of 3 anticodon, and this one also called anticodon loop. Because why it is known as anticodon? Because on the surface of the mRNA some codons are present, and these are called codons, and in tRNA presence of anticodon. After reactions of codon and anticodon, that is converted to amino acid. And the last one, that is the last one, that is the rRNA. Basically rRNA is also single stranded. In some cases RNA, the structures of the RNA just like a loop like structure, these are called rRNA. And in where it will form loop, that means in that region specifically presence of double bond. So rRNA ki kore na, rRNA main functions jeta ribosome formation se help kore. Suppose যদি আমরা রাইবোজোমের স্ট্রাকচারটাকে নোটিস করি, তো রাইবোজোমস আর ডিফারেন্সিয়েট into 4. These are the 70S ribosome, then the 80S ribosome, then 77S ribosome, as well as 55S ribosome. So ribosomes are differentiated into 4 types, and these are 70S, 80S, 77S, as well as 55S. Suppose these on 70S, and after centrifugations it will be separated to 2 form, that is the 30S ribosome as well as 50S ribosome. 30S ribosome and 50S ribosome. These one 30S and these one 50S. After centrifugations, 30S ribosomes again it will be differentiated into rRNA, that is the ribosomal RNA. 16S rRNA, it will be differentiated into 2 parts, 16S rRNA and 21 protein. That means it will be contain 21 number of amino acid. Suppose after centrifugations of the 50S, it will be differentiated into 2. There is the first one, that is 23S rRNA, 23S rRNA plus 5S rRNA. কিন্তু কখনোই আমরা এটাকে মাল্টিপ্লাই করে লিখতে পারবো না. And the next one, that is 34 protein, that means 34 number of amino acid. So in case of rRNA, ribosomal RNA, it contain 16S rRNA, 16S rRNA plus 21 protein, then it convert to 30S, then 23S plus 5S rRNA plus 34 protein, it contain to 50S ribosome, and with the presence of Ca++ ion as well as Mg++ ion, it will be bound with each other, then convert to 70S, and in that condition 10S will be overlapped. So RNA, these are the types of the RNA, and it will be completed.